Hi guys, my name's Catherine and I'm presenting the responding to antigen sections of the biology study design, which is in Unit 3, Area of Study 2. I've put in the learning objectives from VCAR here. As you can see, there's quite a lot to go through, so I'm just going to be giving you a brief overview today. Feel free to pause the video and write things down, as well as go through this in your own time and in a lot more depth if you feel you need it as well. Obviously, key to this topic is what is an antigen. And so an antigen is just anything that's on the cell surface, basically. So usually proteins I've got here, which can then trigger an immune response in the body. So these can come from outside the environment, or they can be formed inside the body as well. And they help the body identify like what's coming from the body and what is coming not from the body. So what needs to be got rid of and what can stay. In my little diagram here, I'm just showing that a cell can have multiple different antigens on the outside, and that there is a surface molecule markers as well. So just a little bit more on distinguishing self from non-self. A really key player here that you need to know about is the MHC markers. And there are two main types of these. So there's type 1 are found on all nucleated cells and they present self proteins essentially. So I've got in the little diagram there the regular cell, and the little red squiggles are meant to present self proteins, and as you can see they've been moved on to the MHC1 marker on the outside so that the other cells know that this is a self cell. The type 2 MHC markers, on the other hand, are only located on antigen-presenting white blood cells. So these cells only include macrophages, dendritic cells, and certain B lymphocytes. And what the MHC class 2 markers do is present antigen fragment to helper T cells to trigger an immune response. So in the antigen-presenting cell diagram, I've got the little green thing, let's call it a bacteria, has been ingested by the APC. And then the digested material is then presented on the MHC class 2 markers to be taken to the helper T cells, which will make a little bit more sense the further we get along in the PowerPoint. So also do note that red blood cells don't have a nucleus, so they don't have MHC class 1 markers. Also in the study design is allergens. So allergens are just antigens that your body is having an immune response to as part of the definition of an antigen, but that they shouldn't be. So for example, like molecules in nuts are very common allergens, which then your body triggers an immune response to, even though obviously a nut isn't trying to kill you, so you shouldn't be having an allergic reaction. Pathogens, though, are disease-causing cellular or non-cellular agents, and these do elicit an immune response, and very necessarily, because they have non-self antigens and they are harmful to your body. So in the little pictures, you can see examples of the bacteria, viruses, and fungi, and in those, you can see examples of what would be considered a antigen, for example. So the squiggles on the bacteria, little sticks coming out of the virus, the spores on the fungi, that sort of thing. Also, I will say that you should know two examples of each type of disease. So like two examples of bacteria, viruses, fungi, etc., and the disease that this causes in humans. So how do we protect ourselves from these pathogens, these antigens which cause disease in our body? Well, there's three lines of defense that we have. The first line are called the barriers, and I've got it written in the human column, but it sort of applies to both here. And their function is to keep the pathogen out of the internal environment. And they are composed of chemical and physical barriers. I'll leave you guys to read through those examples, so feel free to pause. But what I will point out here is that intact skin in the human line, that's a physical barrier, but only if the skin is intact, which is why I've got it underlined and bolded. So you must say that, because if the skin's not intact, then the pathogen could be getting in through a cut, for example, and that could cause you to lose marks in the exam, which is something we don't want. Also in the human colon, lysozymes, please don't get them confused with lysosomes. Lysozymes are enzymes in tears. Lysosomes are cellular organelles. Don't get them mixed up. Don't get their spelling mixed up. You don't want to lose needless marks. And then the plants as well, very similar to the human things, but just the plant versions of them. So when we're looking at the immune system, as in what happens like inside the internal environment, we've got two main divisions: the innate immunity, also known as non-specific immunity, and the adaptive immunity, also known as specific immunity. So non-specific immunity comes into play as soon as the pathogen has entered the internal environment, and non-specific immunity, as the name suggests, responds to this pathogen the same way, no matter what type of pathogen it is. Could be influenza, could be strep throat, could be anything in between, that it doesn't care, it has the same response. And I've got the components there as well. And the adaptive immunity, the specific immunity, it responds to the pathogen after the innate immune system hasn't really had quite a good enough response, and it responds to the pathogen in a very tailored way, specific way, really. So, for example, if you had influenza, your response to influenza is going to be specific to influenza, and it wouldn't work if you had, let's say, hepatitis C, for example. So now we'll just go into a little bit more detail in the innate immune system. I've got the cells of the innate immune system here, so I'd encourage you to pause it and write these definitions down. They're very important, they're part of your study design, and it'll help make the next part make more sense. Also part of the innate immune system is the inflammation response, which you do need to know as part of your study design. And so I'll just go through the steps here. Basically, firstly, injury causes a histamine release from the mast cell. So let's go through this example in the context of, let's say you've had a splinter, it's in your finger, it obviously really hurts. So the injury here is the splinter, which has pierced into the internal environment and caused a histamine release from mast cells, which are then attracted to the site of injury. The second step is that complement proteins move from the blood to the infected area, which subsequently attracts macrophages that move from the blood to the infected area and bind to and engulf the pathogens, because macrophages are phagocytes. The third step then is that blood plasma also moves to this infected area, which causes the swelling because it's bringing a lot of fluid in it. The blood plasma, as you may or may not know, is predominantly made up of water, so it's very fluid. And this causes the swelling, and it causes increased pressure. Like, as you would know when you've had inflammation response yourselves, it gets quite red and swelled up, and you can sort of feel it throbbing sometimes too. And this pressure in your finger is what can help localize the infection there and make sure it doesn't spread anywhere else. And the fourth step also, that platelets from the blood release growth factors and stimulate dermis, which just means skin cells, to divide and heal wounds. And I would encourage you to know the cardinal signs of inflammation too. So when we're talking about the adaptive system, the first thing that we need to know about is the lymphatic system, which is how the adaptive system works. Basically, the lymphatic system runs sort of parallel to your circulatory system. So instead of having like veins and arteries, you have here like lymphatic glands and lymph nodes and lymphatic vessels, etc. And there are some important components to it. So lymph nodes become really important in immune response. Lymphocytes, so they're your T cells and your B cells, also your natural killer cells, which are not part of the adaptive immune response. And spleen is a really big lymphatic organ as well, that's good to know. Also in the diagram, you can see the thymus. It's a yellow dot sort of in the middle. That's where your T cells mature and your B cells mature in the bone marrow. So just keep that in mind. So when we're talking about T cells, there are two types of really important T cells, and they're your cytotoxic T cells, which attack specific viral infected or cancer cells, and your helper T cells, which help mobilize an immune response from cytotoxic T cells and the B cells. Without these cells, you can't really have an immune response at all, and they release cytokines, which are just signaling molecules. So in this case, it's interleukin 2, to activate correct B and T cells. So if we look in the little diagram here, the T helper cell has a T cell receptor, which is what TCR means, on the outside, which I've written recognizes the protein held by the MHC class 2. So then we've got our antigen presenting cell showing this to the helper T cell, which is how the whole immune response gets off to a good start. Well, the adaptive immune response anyway. And then the helper T cell can then stimulate the cytotoxic T cell, also through T cell receptors and the cytokines that we talked about, to then have its own response. So then the cytotoxic T cell can go out and attack viral infected cells. However, the other thing that helper T cells do initiate the response in is B cells. And the important things to know here are plasma B cells, memory B cells, antibodies. Plasma B cells are the cells that produce antibodies. Memory B cells are stored for later, and antibodies are what plasma cells produce. And I've also drawn a diagram of plasma cell here because it's important to know that the plasma cells have loads and loads of rough ER, so rough endoplasmic reticulum, because they're producing loads of proteins for export, and those proteins that they're producing for export are the antibodies. So antibodies, I'll go into in more detail on the next slide, but they help the immune response by clumping free pathogens. So whereas the cytotoxic T cells helped with infected cells, these are clumping the pathogens that are not inside cells necessarily. Important processes in the B cell response is clonal selection, which is when one specific B cell is chosen by a helper T cell, clonal proliferation, when after this occurs, the production of so many B cells from the original B cell happens, and then differentiation, which is when B cells go into both plasma B cells and memory B cells. So then just going over antibodies before we finish, they're also known as immunoglobulins. You see here in the diagram, the red part of them is the heavy chain that looks sort of like a Y, and then the yellow part's the light chain. The bit above the red dotted line is called the variable region, and the variable region at the very top here is where you have your antigen binding site. So this is what binds the free pathogens in the bloodstream, for example. And this bit down the bottom below the red dotted lines are the constant region, which is the same no matter what the antibody is, whereas the variable region changes. But the variable region on each side of the antibody is always the same. Also should note that disulfide bonds join each of the chains as well. And there are a couple of different types of antibodies. You don't really need to know lots about them, but I would know that IgE is the antibody which is important in allergic reactions. Okay, so that's a bit long and a bit content heavy, but hopefully you've enjoyed it, and good luck with your exams.